Yeah, so thank you very much for the invite and for coming out, and yeah, looking forward to giving an update on our highly pathogenic avian influenza monitoring programs, as well as some of the other waterfowl management activities that I coordinate here in the Atlantic region. So just in terms of our updates with what I'm going to speak about in terms of HPAI, so I'll probably refer to highly pathogenic avian influenza as HPI because that rolls off the tongue a little bit easier. But just to put that acronym out there. So we will talk about some ongoing surveillance and results, both at the national as well as the regional level, and specifically to our regional level, talk about our sick and dead bird testing from beginning of April of last year to current. Our live bird testing in non-waterfowl, and our serum and egg testing, as well as our national surveillance strategy that's been rolled out with waterfowl as the primary target group, and our results coming out of kind of quarterly monitoring that we've been doing here since April. So just a little bit about the virus and the background of how avian influenza viruses work. They're classified into two groups. There's a low pathogenic virus, which essentially is on the landscape and has been for quite some time. It infects both domestic poultry and wild birds. However, there's few or no clinical signs, and the transmission is very low. So again, the pathogenic portion of it being low, that doesn't spread between individuals very easily. And it also has a relatively low mortality rate. So this is something that, again, is on the landscape, is out there, and we do detect. But it is not of a conservation concern for wild species or a farm industry concern at the moment. The second version we have on the landscape, the highly pathogenic version, shows high transmission as well as high mortality rates in domestic poultry. The numbers you hear about in the news about tens of thousands of individuals that are euthanized when a detection occurs is because of this highly pathogenic variety. And it is very impactful at commercial poultry and not in Canada anyways, but in the US, commercial dairy programs as well. In wild birds, there's often asymptomatic to fatal infection dependent on species infected in individuals and the viral strain. So basically saying that birds can be on the landscape with this virus. They maybe don't show symptoms, but they can be infected and spreading the virus. In terms of aquatic birds, they tend to be a natural reservoir for low pathogenic avian influenzas as well as high pathogenic avian influenzas. So those are things like our ducks, geese, and swans, as well as terns, gulls, and shorebirds. In terms of classification, when we talk about H1s and N1s and H5s and N7s and those types of things, it's just broken down essentially at the genetic level between the hemagglutinin and the neuraminidase, and we separate those categorically H1 through 16 and N1 through 9. So that just helps us break down which virus is on the landscape and which one is infecting certain areas. With respect to kind of the cycle of the virus, the figure there shows that there's maintenance and diversiveness of strains that occur as the virus repeats itself around wild birds. Kind of in that green diagram there, there's possibility for that transmission between wild birds and agricultural atmospheres where that cycles through those individuals. Oftentimes, because the mortality rate is so high, that strain of virus ends at that facility at that point, or through euthanasias. But there are opportunities to cycle that through again where there's adaptation and evolution. And in very rare cases with respect to the highly pathogenic influenza virus, that carry over into humans. So thankfully in Canada, to this point, we've had one exposure. I am not a public health doctor or anything that way, so can't really speak too much to the avenues that way, but we have had a case of a teenager in British Columbia being infected by the virus, and last update I think on recovery from that, so that is great. But there is the potential there for influenza viruses to move out of wildlife and into humans. With that, the overview of our surveillance streams and how this applies at a national level. Sick and dead bird surveillance is led by the Canadian Wildlife Health Cooperative. The location for that here in our Atlantic region is at the veterinary college in Prince Edward Island at the university. There, between the Wildlife Health Cooperative and the provincial or territory governments, they are the lead agencies for receiving reports as well as collecting sick and dead birds and analyzing those individuals. So for our area, birds that are collected are sent to the Canadian Wildlife Health Cooperative, analyzed for what might be the reason, and testing and confirmation of that mortality after the fact. Collecting agencies retrieve birds and deliver those to the agencies responsible for sampling. So again, provinces and territories are taking calls of sick or dead birds here in the various maritime provinces, putting those forward to the Canadian Wildlife Health Cooperative, and they're analyzing those. And if those reports meet certain triage criteria, samples are entered for avian influenza testing. So there are other reasons that wildlife is passing away on the landscape. Not every dead bird necessarily means avian influenza virus. So those birds are looked at more closely and determined after the fact what that mortality event's from. With respect to live bird and active surveillance, that's being led by Environment and Climate Change Canada, and most specifically here in our area by the Canadian Wildlife Service. We take our opportunities where we can for sampling apparently healthy birds, primarily through our existing programs, so our waterfowl banding program, which I'll talk more later on tonight, but that's a key program we use for our active live bird monitoring, as well as some targeted efforts through hunter harvest and other programs that allow us to kind of get access onto the landscape through citizen scientists, and other volunteers. We collect those samples through these identified opportunities. So again, whether that's banding or hunter harvest samples, and those are sent directly for avian influenza testing, because that's the purpose of that program, is to be monitoring for avian influenza. So all the samples we're taking are being sent forward for avian influenza testing, and results will be coming up here in a minute. In terms of our kind of overarching program response to avian influenza, we have 4 streams of which we're assessing this, first being waterfowl swabbing. So the goal of our waterfowl monitoring is to optimize detection of avian influenza viruses to maintain current data under our One Health framework and inform risk. And we'll get into kind of the breakdown of the numbers, but essentially we're sampling enough waterfowl year round to detect a 1% prevalence rate in waterfowl on the landscape. So it's quite a good level of effort out there, to appreciate if there's 1% on the virus. There's 1% occurrence in the virus on the landscape. We're sampling enough birds to be able to detect that. We're strategically sampling key reservoirs to maximize virus detection by existing bird in hand programs. So I mentioned earlier that ducks, geese, and swans are one of these reservoir species groups, as well as gulls and terns and shorebirds, and we're actively sampling waterfowl, but also opportunistically sampling other wild birds as well. We're combining some of this testing with blood samples as well. That helps us to inform susceptibility in wild birds, gives us an antibody level, so whether that bird's been exposed in the past and survived, as well as survival rates. And we're again targeting this approach in priority species, primarily waterfowl, but also some that are a potential conservation concern, given that gulls, terns, and shorebirds do have some species at risk associated with those in the Atlantic region. We are trying to sample some of those individuals as well, and species that might have more of a domestic animal or human interface. We're also looking at egg antibody analysis. This helps us inform the susceptibility in wild birds, previous exposures, and survival. So if antibodies are being deposited in eggs, that's showing sort of a adaptation to the virus and providing that antibody exposure kind of in the egg that might make those young a little less susceptible to the virus moving forward. So being able to monitor that sort of from first exposure of this novel virus in North America moving forward is a timely and important thing to be doing. This is a minimally invasive sampling technique through existing infrastructure of ECCC programs tied mostly to contaminants. So there's established programs out there looking at things like heavy metals, plastics, petroleum products, and that type of thing being deposited in eggs. We're just taking an additional sample essentially from those eggs to test for antibodies of past exposures to various AIV viruses, and then also mortality assessments. So assessing impacts to affected populations, and then doing some population monitoring and modeling to determine impacts on those priority species. So I'll talk about northern gannets and common murres a little bit later on, but two seabird populations that were heavily affected in the first kind of exposure year in 2022. The One Health approach that I've mentioned and will mention again comes through the World Health Organization, and this One Health approach aims for integrated and unifying approach that aims to sustainably balance and optimize the health of people, animals, and ecosystems. So kind of taking our monitoring at a level where we're looking at overall animal health, but how that impacts human health and overall environmental health as well. So in terms of national surveillance, this is showing detections of high-path H5 avian influenza from all sources, so whether that's direct mortalities, harvested individuals through hunter-harvest surveys, and live bird surveillances since November 2021. And you can really see that we had a really large uptick in April and August in our region specifically, but as well as nationally, and that was about 4 months after the virus was first detected in Newfoundland. Leading into the next year, 2023, again, springtime seems to be a point at which the virus sort of resurfaces. We had a relatively low occurrence during spring through summer in April to August, 2024, and 2025 is yet to come forward, so I don't have any information to show there. But basically what this is showing us that the virus hit the continent. We had a huge upswing of virus exposure and mortalities associated with that, and now that that's been on the landscape, there has been some kind of prevalence that's occurred. Species are able to get through that and maintain, but that doesn't mean that the virus has necessarily disappeared from the landscape. In terms of our regional surveillance, looking at sick and dead and live birds, since April 1st, we've had a little over 1,200 samples submitted. Of those, 31 were positive for high path, 18 positive for the H5. This matrix positive essentially suggests that they can't tell whether that's high path or low path without further genetic testing. So there's been 61 positives that have had to go on for further testing. The number I think that's worth looking at the most is the almost 1,100 that were negative, and results pending of some of that further analysis. So we're waiting for 13. You can imagine we have one lab that's in Winnipeg that can do the genetic testing in Canada, and so the volume of samples coming through to that lab has quite a bottleneck out of that institution. So we do have to wait sometimes several months for sort of the genetic blueprint of which strains of viruses there are, but the primary testing, we have those results back within a couple weeks, which is really great. In terms of surveillance and where positives have come from, up until recently here, Prince Edward Island had 63 specimens submitted for testing. 6 were positive for high path avian influenza. The springtime, there was 4 red foxes and 1 striped skunk. In the fall was 1 greater black-backed gull. New Brunswick, up until we heard, I guess today or earlier in the week, there was no positive individuals, but a recent red-breasted mergansers tested positive. Nova Scotia had 25 positives out of their 210 that were submitted. Again, these are dead individuals. So there's been several species of gulls. So 5 greater black-backed gulls, 4 herring gulls, Iceland gull, and then a couple of fulmars, common raven, American crow, common tern, red fox, all seabirds except the 1 black-backed gull and the 1 common tern were from Sable Island. Sable Island's this lovely catch-all drift net of birds that perish at sea that help us to monitor across species and as well as across time. I guess it's hard to refer to things as nice parts of viruses, but the nice part of avian influenza virus is that it's quite acute, so the individual dies quickly, which means that a lot of decay isn't necessarily happening associated with that virus. So if birds are found early enough, they can be tested even after death, and the virus will have kind of preserved itself in there for a quick test. Newfoundland, there was 97 specimens submitted, 11 positives came from that, and most of those are from gulls. In terms of our strategy through the year of the surveillance, again, we're looking to some models to calculate our sample size needed to determine minimum sample sizes. So again, looking to assess a 1% prevalence rate on the landscape. We took a flyway adapted approach and applied that as functional ecological boundaries. So we know that there's 4 migratory flyways across the country, and we broke our targets for each of those flyways, and then broken down by provinces within those as well. And then trying to do that within the flyways equally across seasons, so that again we're getting this detection rate of 1% spring, summer, fall, winter. And then using a risk-based allocation of samples at a provincial territory scale based on eBird abundances to try to detect increase spatially over time in that high-risk areas, and again discern species of interest and concern. So in terms of seabirds, we disproportionately impacted in 2022. Again, I'll get into the numbers in a little bit. We've had continued regional surveillance since then, and directed and opportunistic effort has been reduced a little bit since 2023, and that's mostly because post 2023 we haven't detected a lot of HPI in any seabirds, which is wonderful. Approaches to that monitoring coming from swabs, from blood showing antibodies as well as past exposure, and then looking at eggs as well for antibodies being deposited in eggs from the female. In terms of live surveillance for seabirds and shorebirds, this is all the samples here coming out of this year. We had 13 different species, 276 samples. A low path, again, the one that's been on the landscape for quite some time and doesn't have major impacts at about 0.7%, and no positive results for high path, which is great. Further in the seabirds, again, blood allows us to look at the serology, which is essentially the past exposure of avian influenza viruses to these birds. This was a collaborative project between ECCC, Canadian Wildlife Service, the Canadian Food Inspection Agency, and Memorial University, and we were collecting blood. The top image is of what's called a Nabudu strip. It's essentially a cotton swab that pulls blood up, and once dry, stores all the information that they need for analysis in that strip, and we can send these strips from across Canada to these labs for analysis. 2024, we had a bit more of a focal study on gulls on Kent Island, looking at interaction of the virus, antibodies, and contaminants in diets. And so just being able to have all of these projects going on at once and adding antibodies as another test was a great opportunity to get understanding on the landscape of what's gone on in the past. So from sampling of this focus study, we had 7 or 23% detection of low path in herring gulls, and about 55% or 6 great black gulls coming back. The results pending is coming from the genetic analysis of that antibody work to determine whether any of those antibodies are from the high path avian influenza virus, and again, that's got to go to Winnipeg, and a bit of a bottleneck there. So we're still waiting on that information. Looking at seabirds, again, we're collecting seabird eggs to look at other contaminant information, and just able to use that opportunity also to look at antibodies being deposited by females into those eggs. Again, another collaborative project through those institutions. You can see here in 2024, we had a great opportunity in combining some of this surveillance through some of our other programs. We conduct aerial waterfowl surveys in May and June across all the Maritimes, Newfoundland and Labrador, and combining some of that helicopter effort allowed us to get just different colonies and do a greater collection, which was great. So we had approximately 52% of all the samples collected coming from our region, which is great. And what that showed coming out is that essentially after the main exposure time in 2022, there was a lot of antibodies in bird eggs in 2023 and a reduced amount in 2024. And what that tells us is that the amount of virus being exposed between 2023 and 2024 was reduced for those species that were monitored, but the prevalence had been quite high the year before, and that's coming out of individuals that would have lived. So again, we'll talk a bit more about gannets and common mers, but heavy die-off in that first exposure year in 2022, which you probably all remember. In terms of our national live surveillance strategy, again, focus species are dabbling ducks. They're a key reservoir for avian influenza viruses, and they often are the drivers behind transmission and spread. These birds undergo long distance migrants across great areas, and they also concentrate in relatively high numbers. So being a reservoir for viral spread is something that ducks are good at doing. They host a large diversity of virus genotypes and are prone to co-infection, so that allows for kind of a boiling pot type idea of mixing different strains together and potentially some evolutionary change of the virus over time. And we're using existing monitoring and banning programs to help collect these samples. So I mentioned that we took a flyway approach, so there's a Atlantic, Mississippi, Central, and Pacific flyway across the continent, and within that we broke our samples down based on numbers of birds, concerns of other species associated in those flyways, and then broken down essentially to individual provinces based on abundances across a time series. So basically for us, we're looking at roughly 250 samples every quarter, broken a little bit differently across the provinces as and where we can, to roughly just over 1,000 samples in the past year. So within our spring surveillance, it's a bit of an awkward time for us to be on the landscape. Ducks aren't just a kind of day trip opportunity necessarily always to go and catch and monitor and sample, so we had a bit of a reduced sampling spring effort. Nova Scotia, we were able to get out some sampling on a lot of local ducks in and around the HRM area, and we found just one case, or 33% of low path in those birds, no high path tested, and then we ended up catching 50 spring migrant geese in Prince Edward Island, and none of those tested positive for any virus. So of the total samples, we had roughly 2%. So again, we're looking at a 1% prevalence rate for sampling, and even not hitting that, we're able to meet that kind of detection level, which is great. In the summertime, we have our regular scheduled banding program on the landscape, which I'll talk a bit about more later, but we essentially handle almost 4,000 ducks through the summertime and early fall, so getting to our sampling quota across most of our provinces was easy to do. No high path, and you can see the prevalence rate there for low path. So again, still being able to detect at a relatively low prevalence rate based on our sampling size. In the fall, we combine partially through our banding program as well as hunter harvest samples. And so we started to see a little bit of an increase in high path on the landscape. And this is really kind of an expected result. Essentially, ducklings that hatch that maybe haven't received any antibodies through the eggs are novel, this is a novel exposure to them, so they've not been exposed to virus on the landscape. And through banding programs, we often are handling a lot of hatchier birds and trying to do our best to sample only hatchier birds, because they give a better understanding of what the virus is doing in the population, because they haven't been exposed and maybe don't have those antibodies to fight the virus off. So seeing those numbers is something that is not unexpected for us, and an overall prevalency rate of 6% is within levels of which we've detected before. Wintertime, we've had a bit of an uptick here, sampling through to the 13th of February. A lot of our birds, at least at a higher prevalency rate there, 35% in Nova Scotia, being detected. Most of them out of our HRM work, but some birds in Truro and a couple of birds in Cape Breton as well came back positive for high path, and an overall prevalency rate there of 22%. So again, this time of year, birds are coming out of a relatively stressful time period. It's been winter time. Any birds that are still here are quite concentrated into remaining open water and remaining food areas, and being stressed, birds that maybe are carrying virus start to shed that virus, and it's something that spreads across birds a little bit easier. Now that we kind of have spring in the air, opportunities are going to open up for those birds. Other wetlands opening up, they're going to start migrating, and so that concentration of birds is going to spread out and open up. So transmission typically drops through spring and into summer, and we typically don't see transmission happening again really until concentrated populations start migrating in the fall. So just to summarize all of that, in spring, again, we're out flying a lot of surveys. It's a bit harder for us to actively run a month or two worth of traps, so we were unable to achieve our sampling targets, but trying to do a greater opportunity to this coming year, successful through the summertime in New Brunswick, Nova Scotia, and Prince Edward Island. We don't have any banding opportunities currently in the summer months in Newfoundland, but looking towards some options that way this year. So excited to get that going on the ground. In the fall, again, successful across the region with our sampling effort. In the winter, we were successful with our waterfowl work, but we've been trying to do a little bit of MERS sampling through hunter harvest work in Newfoundland. Newfoundland is the only province where MERS are harvested, and gives us an opportunity to look at long-term exposure and previous exposures through the serology work and the blood work of a harvested species, but also of a species that took quite a dramatic impact to population locally in Newfoundland in 2022. In terms of additional surveillance, in this past year, we did some paired sampling, both blood and swabs from Canada geese. Pairing this up, I forgot to mention, but I should mention, the swabs allow us to test for active virus that's currently in the bird. So virus is shed through the mouth and is shed through the cloaca. We take a swab from each of those locations, and that gives us an idea of whether there's active virus, whether it's low path or high path in the bird at the time. Whereas the blood and the serum analysis allows us to look at past exposures. So if there's antibodies, essentially like a titer. I don't know if anybody here works with livestock, but if you've had your rabies vaccine and you go back again, they check for your titer, which is just showing an antibody prevalence in your blood. That's essentially what our blood work does for some of these birds. So antibodies in the blood for high path means that bird has been exposed to high path, has lived through that, and just shows the antibodies in the blood. But the swab paired with that might show that it's not actively spreading virus at the current time, if that hopefully makes sense. In terms of our paired waterfowl sampling, we did some bleeding of birds during our airboat program. I'll talk a little bit more about that in a minute. But we were able to sample 55 American green-winged teal from Prince Edward Island in August, and then we did almost or just over a dozen birds in September in Nova Scotia. Of these birds, while the numbers are relatively small compared to our swabs, we want to be testing only adult birds that have had at least 1 calendar year to be exposed to the virus. We know from past work that birds that hatch in a year typically have not been exposed, and they wouldn't have time to have been exposed to the virus, recover, and then be showing antibodies. So bleeding young birds doesn't provide us any information. So we need to be sampling adults, and we don't always get opportunity to catch those with the airboat. And I mentioned, not gonna let me do that. Sorry, I'll try not to do that this way. Now I'm touching too many things at once. That's where technology is always fun, but not fun. Some of our waterfowl sampling, again, from hunter harvested birds. hunters would harvest a bird. We gave them a sampling kit, so they would be swabbing the mouth and the cloaca for active virus, but also taking one of these Nabudu strips and collecting blood from that bird to then dry that out, and then we're recovering those and sending them for analysis. So it gives us an opportunity to look at active virus on a time period outside of our banding efforts, as well as being able to look at past exposures. So it's been a really good collaborative effort across the Maritimes and Newfoundland with hunters on the landscape, which has been really great. So talked about all of this kind of One Health approach and the importance of that, and how we've taken that approach in Canada, and it's kind of nice to get a published acknowledgement of that effort. So this study was published recently in a biomedical journal looking at viruses and things, and complimented Canada on its exception of being a great program for monitoring avian influenza on the landscape. Taking this One Health approach has really been a great way to look at impacts of wildlife, but as well as domestic animals and human interactions that way. So it's kind of nice sometimes to get an accolade and an accomplishment out there from other folks. So feeling very confident in our approach and how we're proceeding with monitoring high path on the landscape, and it's just nice that get the tip of the hat to that as well. With that being said, this undertaking is ginormous. There's a lot of people involved in bringing this all together. Obviously a lot of moving parts and a lot of people putting a lot of effort in this sampling. So thank you to all of those folks. In terms of where this goes, when it first hit the landscape back in 2022, we had quite significant die-offs of northern gannets, common murres, as well as common eiders. There was a lot of public concern. Those of you remembering that time of gannets washing up on the beach in quite high numbers, kind of out towards Cape Jury-Main, definitely in the North Shore of Prince Edward Island was quite a unfortunate sight to see if you were there, just with the numbers of gannets dying, and then Newfoundland having similar gannets as well as murres washing up on shore. Because of the impacts to some of the larger eider colonies in the Gulf of St. Lawrence, we requested a voluntary reduction in harvest of brown eiders. So females are the ones that reproduce. They saw roughly 5 to 15% mortality in adult females across some of these colonies, and so there was a request going out to Canadian hunters as well as American hunters for a voluntary restraint on brown eiders to just sort of help that population through the hunting season. We didn't feel that there was a significant impact where hunting needed to be reduced or eliminated that year, but kind of a hands-up help in reducing female mortality we felt was something that could be done on a voluntary rate, and something that we saw quite broad application across the hunting community, which was great. There's been a lot of collaborative research efforts underway as well to further refine and communicate the estimates overall of the total wild bird mortality in Eastern Canada. So with that being said, we know from gathering carcasses that there was about 23,500 dead northern gannets, but thousands not accounted for. One of the things that we looked at, some of our science group looked at particle modeling through drift. So if you can imagine the tides and the currents off of the coast of Newfoundland and as well as in the Gulf of the St. Lawrence, if those birds were not brought into a current stream or a tidal direction that would bring them to shore but actually push them out to sea, people wouldn't ever know whether those birds had died or not. So they actually used small particles with transmitters on them to look at where they moved in time relative to where the colony was and to where birds were being found on beaches, and that those results are still coming in, but it's estimated that both for gannets and for murres that potentially thousands of individuals were not recorded because they never would have drifted to shore in the first place. the estimation for the eiders, not lucky for the eiders, but kind of lucky for us, and our understanding is most of the birds died in the colony at fresh water sources. So they were going to those sources. Infected bird was drinking or defecating in the water source. Other birds are coming, and there was freshwater areas on these islands with dead females. And because these eider colonies are long-term monitored with leg bands, they can get an estimate of total number of females that would have died based on doing some mark recapture analysis on dead birds. So the positives that come out of this are the breeding colony surveys after 2022 showed that they're increasing in numbers of breeding individuals. Anticipate at this time, a recovery of those populations, specifically gannets and murres for this, but it'll take time. These birds are long-lived. They can take between 5 and 7 years to reach sexual maturity, and then you think of the cohort of adults that died. That's going to take quite some time to replace. The silver lining, though, is that of the birds that did survive and those that are coming into that adult cohort, the competition there on those colonies is now reduced. So that opportunity when they get there to breed is greater for those individuals, which might help kind of bump up that reproductive effort, because that density dependence and impacts of density on colonies is less now than it was before. And in theory, those that survived are the ones that maybe can fight this virus moving forward as well. So again, sort of silver linings that come out of a pretty negative story. Some of the other things that we get that's a positive out of negative stories is the information that we learn and the scientists learn that they can share through publications to others to understand moving forward how best to assess and then also react to these. So this is just a list of publications that have come out from the work that Environment Climate Change Canada through our Science and Technology branch and other partners have done, and a bunch of great science out there. If you've got more interest in that, you can reach out to me afterwards, and I can get you some of those. In terms of HPI and wild bird human interactions. There's lots of wild humans too, but we're more interested in the wild bird and human interactions. This is pulled off of our ECCC Canada.ca Avian Influenza and Wild Birds link. If you go to Canada.ca and search wild birds, it'll bring you to a sort of a breakdown in fact sheets that you can scroll through. So topical now is obviously winter bird feeding and feeding birds in your backyards. The species that come to our backyards typically are not ones that are susceptible to HPAI. There are a lot of birds that are reservoirs, so we've talked about waterfowl that kind of hold the virus and share the virus. Many other birds are an end source, so if that bird was to get sick, that bird dies, but doesn't necessarily spread the virus on to another one. So a lot of our backyard birds and feeders typically are not having waterfowl come to them. If your backyard feeder has ducks, geese, or swans coming to it, I'd probably recommend that you don't continue to feed, because that potential is there with that reservoir species there. But if you maintain appropriate cleaning regimen of using 1% bleach in water and washing your feeders every 2 weeks, at this point it's considered relatively low risk to be backyard feeding at this time. Again, if you are in a place where waterfowl are around, downtown here next to the retention ponds or the waterfowl park, it's maybe not the best choice to do. But if you're in the woods and away from those species, the opportunity is pretty low risk. There's lots of information online about timelines and recipes for making disinfectants, but if you keep that up, that's going to keep your backyard birds safe. And with respect to individuals and finding birds, that is something that happens. I had mentioned that it's the provincial level that's the primary reporting mechanism for finding sick or dead birds. In Nova Scotia, there's a toll-free 800 number to call, and in New Brunswick, it had asked that you contact your local DNRR office, which is DEP for us here, and then they have a triage that they'll work through based on numbers, where you are, where the bird is, what type of bird it is. If it's a gull or a duck, it's probably hits higher on the list than something like a sparrow or a songbird coming through during migration in the springtime. But again, if you want to read larger size font or have more questions about the links there, just search Avian Influenza Canada.ca, and that'll bring you to our information page there. So with that, I will just keep going, I guess, for the sake of time, and we'll do questions all at the end, if that works for everybody. Or do you want to do, how's everybody feeling? I know it's a little bit dry. All good? All right, well, I'll jump into just quickly going to talk about our preseason waterfowl banding. Again, we're using this as our real mechanism for monitoring avian influenza on the landscape. I'm involved with 3 really cool GPS tracking projects, and I'm just going to give some quick updates on that we're doing with North Atlantic population Canada geese. I've got 2 projects going with mallards and American black ducks, and just finished up a project tracking American woodcock. So preseason winter banding, we conduct this to monitor waterfowl populations. Bands are deployed. Hunters that harvest a banded duck report that number online, or there's a 1-800 number, and then that gives us understanding of what harvest rates are. We can apply that to our population models and get an understanding of how many birds are being harvested, what the population needs to be to maintain that harvest, and those types of things, as well as where these birds go once they've been banded. So in the preseason time in August and September, we're banding these birds either in the wetlands that they were raised or during their migration in fall. And when bands are returned by hunters, we get the date of where that bird was, or that location, as well as the date of where that bird's been harvested. So some of our birds are harvested quite locally to where they're banded, and others are harvested in South Carolina, down to Floridas, and we've had some teal come back from the Caribbean. So we do have quite a broad range of birds being harvested across the Atlantic flyway, and it leads to a lot of really interesting information. But it also allows for monitoring avian health. Specifically now, we're looking at highly pathogenic avian influenza virus, but about 5 years ago, we were doing similar swabbing for low pathogenic avian influenza virus, just because, again, it's on the landscape, and having an understanding about that is important. So this is just an image from our winter banding program that we were doing in February down in HRM. We're catching ducks and processing a bunch of them, and that's just us swabbing again for active virus. In terms of our locations, they probably don't show up too well, but we have a banding station in the Codroy Valley in Newfoundland. We have a couple of stations locally out of Sackville here. One's at the Shepparton National Wildlife Area, and we have a coastal station that runs from essentially Shediac to Cape Jury, Maine. We have a station that runs along the St. John River outside of Fredericton. We have a station up in Bathurst and a station down at Church Point, run by University of the St. Anne, which is great. So I'm trying to get a broad area of coverage, so getting multiple different populations of ducks coming together, and trying to disperse our sampling effort across the landscape. If I can make this work, I will play this and it'll explain itself. But we do run an airboat program at night. If this works, that'll be great. I'm going to skip that and hope that that's okay, because I'm sure CTV News is safe to go to, and this hopefully just plays for me. Maybe? If the hamster's too slow, you'll just listen to me and... Alright, so we'll do that.
Stay up to date on all the latest technology and new model features, and have the tools and training for when your vehicle is in need. Certified Service, the service your vehicle deserves.
Now we go behind the scenes of a fascinating initiative, the Canadian Wildlife Service's duck banding program in the Maritimes. As CTV's Josh Smith shows us, it reveals many facts about our feathered friends.
This time of year, waterfowl biologist Ted Barney and his team with the Canadian Wildlife Service in Sackville, New Brunswick, are tasked with banding over 3,000 ducks across the Maritimes. They collect a lot of species right next door on the Tantramar Marsh. which is a popular home to 6 to 8 different breeds of ducks.
We do waterfowl banding to help estimate populations, rates of harvest and survival, and use this information to help set bag limits and seasons for the fall regulated waterfowl season.
In conjunction with the moon phases, collecting the ducks is done by using a water boat powered by a Corvette motor. They set out to a wetland site before dusk, then wait for the sun to completely set. With nets and spotlights, Barney says this is the most effective and efficient way. And then they head out in the boat. To witness it, it looks like a scene from an alien invasion movie.
The airboat allows us to get into multiple wetlands in an evening and be effective to get as many birds out of those wetlands as we can, kind of one night rather than putting in a month's worth of effort. And basically the darkness of the night allows us to use the lights on the airboat to blind the birds. They can't see a horizon, so they don't fly, and then we can use our dip nets to scoop them up, bring them back to shore, and then we band them.
Once collected, Barney and his team assesses each duck for sex, age, species, and they even test for diseases that goes into a national database.
Highly pathogenic avian influenza has been on the landscape in North America for roughly the last 3 years. We have a monitoring program to look at background rates of the virus, and this technique allows us also to catch those samples that we need to take in order to be monitoring waterfowl health.
Then they are banded and set back out to their natural habitat unharmed.
There we go.
This process may seem noisy and invasive, but Barney says it's been practiced this way for centuries, and the animals are handled with care.
Very much appreciate that it is not quiet. We do have everything on the boat to the best of ability to reduce that, and that information coming from this is really critically important to what we do at the Canadian Wildlife Service in terms of managing these populations. So we have protocols in place that are set to an animal care standard that's followed. It's assessed by a committee that includes veterinarians.
Barney wants people to know that anyone can get involved with this process. If you find a duck with a band, you can go online and report it and get the details on that specific duck. Josh Smith, CTV News, Sackville, New Brunswick.
Cool. All right, now I got to get back and hope that all works too. All right. So essentially, airboat, we run that at nights in the dark cycle of the moon phase. We do a lot of that work around the border region here just because of logistics. We do make a trip to PEI at least once a year, and then we do a few nights in Fredericton. We're trying to expand that opportunity as much as we can. So if you have some wetland area that you think would like to have an alien invasion come summertime, let me know, and we'll take a look and see if we can get our boat in there. So the main part about our banding really plays into harvest management and population monitoring. We have certain species that have different population models that require minimum numbers of bands to be put out in a year in order to maintain that understanding. The primary one for us is American black ducks. We need to collectively, before the season starts in North America, band at least 2,000 American black ducks in order for us to maintain our modeling as well as the monitoring of that population. And within that, our region takes on roughly 50% of those birds. Obviously, Atlantic Canada, for a coastal species that's on the eastern part of the world, we are a stronghold for that species. Quebec kind of picks up the other part of that lion's share of that sample, and Ontario kicks out a few too. So between our numbers and the other two provinces, we're over 2,000 a year, which is great. But that's the species that really drives our preseason work. Some of the other species that are key are mallards, green-winged teal, wood ducks, and ringneck ducks. It plays into some mixed species modeling that's done to help bring forward hunting season length as well as bag limits. And so banding's really important for setting all of that season level effort. And so the roughly 3,700 birds that we did this year, you know, it is down a little bit over last year and over long term, but we're hitting the kind of the short term averages and above that, which is great. and that just means we're getting enough bands out to be able to continue modeling those populations in that harvest. In terms of winter banding, we run winter banding primarily for assessing survival of certain ducks, mallards and black ducks specifically, but it also provides opportunity for us for some of the research work that I'll talk about here in a minute, as well as our avian influenza sampling. So this past winter we did quite well. Tough winter conditions mean ducks are really happy when corn's coming out to them, so they're a little bit more interested in going into the trap. That was a busy morning for me. We had just over 45 ducks in that trap. That was that lovely week in February when everything was -20 with the wind chills. So I didn't put the picture in of my waders standing by themselves because of the ice, but it was that kind of cold day. So lots of fun, but birds are in good health. It's all great that way. The neat one that shows the effect of winter for me is the 3 greater scaup that left the water, walked up on shore, and ate corn in a trap, right? Greater scaup are not a walk on shore kind of duck. They typically are eating mussels and things out in the ocean, and these ones were hungry enough that they needed to kind of walk up on shore. So it gives an idea of what the winter's like. Again, leads to that understanding of figuring out survival at this time of year is critically important, and leads to a couple other projects I'll talk about here in a second. But we had a really good winter banding year, which was great. Mentioned that I'm doing some neat GPS work on different populations. One of those is the North Atlantic population of Canada geese. We're deploying satellite GSM GPS transmitters, which I think I'll have a better image of here in a second, but it's deployed on a net collar with solar panels on it, and that net collar sends a signal every 10 minutes for its GPS location to a satellite. That comes back down to the collar, and then the collar actually uses cell towers to download data. So I essentially get a text message from each of these net collars while they're in cell range, and then I can use this website interface to map out where those birds are. We started this project in 2023, deploying these net collars, and this is the wintering areas down here that these birds go to. So they really concentrate in the New England states. They migrate up through the maritime spring and fall, and then we deployed a bunch of collars in 2023 in Labrador, and they went back there to those areas that we caught them in. They are very strong philopatric individuals, so they have a homing sense back to areas that they've been successful nesting, and essentially all the birds that we deployed collars on the year before went back to the wetland system that we had found them the year before. So really a neat way to see where the birds are going, and some of these super unique migration trajectories that happen when they get blown off course, which is really kind of cool, and things you don't appreciate without seeing it kind of more or less in real time. In 2024, we had 2 field programs. The first was in April in Prince Edward Island. In total, we caught 265 geese. Of those, we put 19 net collars out, and we put out 102 nano tags. So I think there's been presentations at some point here talking about the Modus wildlife tracking system with shorebirds. They put on little radio transmitters on the shorebirds. We did something similar, only in a backpack style with the geese, and released those and used the Modus tracking system to track those geese as well. Our spring collar movements were sort of typical of what we expected, but there's also some geese that come from Greenland and use the Maritimes to migrate down to the wintering areas, and we happenstancely, I guess, caught one of those Greenland birds, and we deployed it on PEI, and you can see its migration track to Greenland, and then way up here, this is roughly the Arctic Circle, right about there. So that bird left and nested above the Arctic Circle, which is super, super cool. In terms of breeding effort, these net collars have an accelerometer in them, and it's essentially a sensor that monitors 3-dimensional movement, and you can look at how that movement changes over time to infer different behaviors. So they've put them on captive birds and followed them through a reproductive cycle to see what happens when a goose is laying an egg, what happens when a goose is nesting, what happens when a goose is tending to its goslings, what happens when they're walking and feeding. that type of thing. And essentially on the graph here, these red dots are how much the bird moves in a day, and the blue dots are the relative distance that bird moves within the day. So if you imagine I could walk around this room, and I could walk around this room all day long, and that red dot would kind of do this. But if I was moving and all I did was stand right here, there'd be some movement, but that relative distance essentially becomes zero. And when a goose sits on a nest, she is essentially not moving at all. They get off, they defecate, they might take a drink of water, eat a bit of grass, they get back on the nest. And when these lines separate significantly, this tells me the number of days that that bird sat on the nest, and if it's roughly 28 days, we can tell from that data that that bird was successful in its nest. Whereas in the same time period, if those lines never really diverge, that shows us that that bird either didn't nest, so it deferred, or there might have been something during early egg laying that caused that bird to not finish. So maybe the nest was predated, some other kind of event happened where they decided not to nest. So from our 2024 callers, we had 20 that sent us data at the time. 14 nested, 10 of those nested successfully. So again, their data looks something like this. They had roughly 28 days in a row of when they weren't moving very much. 4 failed during incubation. So that would be something like this line stops after 4 or 5 days. We had 12 that deferred, so it would have looked something like this, where they didn't actually attempt to nest at all. And then at the time that we've run this analysis, we had 3 that hadn't come back and checked in yet. So again, the data comes through a cell tower, so if they're not in cell coverage, we can't get the data. Average nesting date in Labrador is between the 15th of May and the 15th of June. That bird that we caught that went to Greenland, she successfully nested. But she did so almost a month later because of the distance she had to go and how much reduced succession is or progression of spring is that far north. So some really cool information that we can get from these net collars.
Ted,
Yep.
do you ever get a graph where it might have failed in the early and then re-nest?
Not with geese. So
Okay.
Geese are what we call a capital breeder. They have to take all their energy they put into eggs with them up to the breeding grounds. And when they've put that energy into eggs and that nest was to fail, they don't have the ability to build that energy back up and then lay a second clutch. Whereas the ducks that I'll talk about next, they show up and they lay kind of the number of eggs that the habitat will allow them to grow at that time. And if they fail, they have time, because they're getting their energy locally, to be able to nest again.
Oh, cool.
So the second part of our 2024 field season, we were in Newfoundland, and I had 21 collars left to deploy, 20 nano tags, and we caught a total of 144 birds total. These were captured in brood flocks, so we find a flock with a helicopter, and we can see that there's goslings there. We look around that flock for an area that's relatively dry, and we drop the helicopter down. The catch crew gets out and sets up a giant U, and then I stay in the helicopter with the pilot, and we use it kind of like a border collie to herd the geese into that U. And once they're in the U, we bring the wings around, and that captures the geese, and then we land the helicopter and then process them from the ground, which is kind of a lot of fun. I won't lie, it is a lot of fun. Anyhow, our fall movements of birds, you can see this is where we were here in Newfoundland deploying these white color transmitters. They, at the time, was making those maps. We're moving down. We get most of our birds coming through the Maritimes, and then as migration proceeds, they end up in the New England states. This Greenland bird took a very different path down, so if you remember back, it did this in the springtime. In the fall, instead of a more direct approach, it hit Southampton and bounced along, crossed over into the upper stretches of northern Quebec, and then bounced down through Quebec and ended up spending the winter in suburban Philadelphia in a backyard stretch of ponds, which I find amazing that a bird that has been that remote all summer is so urban when it's not, but it's a great survival strategy. Not to say that it chose us for sure, but because once she got south, we could look at her data and see that she nested and was successful. This sort of protracted migration coming south might be explained by having 4 or 5 goslings that are still growing, needing to build that endurance to migrate. And instead of hitting a straight line across that giant chunk of the Labrador Sea, she hit Southampton, bounced along, gave them time to continue to grow, recover that nutrients from migrating, and then make a couple like kind of leapfrog steps heading down. And then right now, we've got relative current locations. I should have updated this because we've got a whole bunch of birds moving north in the last 3 days, but that's where birds overwintered. From previous work, we know that birds coming out of Newfoundland are typically the ones that had stayed sort of in southwest Nova Scotia, and I know the colors are hard to see, but these white colored pins are from Newfoundland birds that we marked this past summer, and they did do that, and the bulk of the other birds are down, again, New England states. So some really interesting information that's going to come from this project in the years to come. Got another one here that hopefully this will work while I'm talking. So we've been, for the past 3 years, putting backpack transmitters on mallards and black ducks. It uses the same technology as the net collars do. So it's a transmitter that takes GPS points, conveys that through cell towers, and gives us the same accelerometer data. So again, we can infer things like nesting, incubating, brood rearing. This year we deployed 16 transmitters. The projects for each of these species total is roughly 500 transmitters each. We're also doing some geolocator work on mallards, and geolocators look at light-dark phase cycles to determine how far north that bird goes, and it's just a bit of a lower impact transmitter to put out on a bird, but we have to catch that bird and remove the geolocator. We put out 15 last year, and we recovered 2 from 2024. So it's not necessarily a great recovery rate for something like a duck, but it works really well for seabirds. So there's been a lot of really cool geolocator work done on seabirds from other projects. Now this one hopefully is going to work too here. Get this to go. This is the spring migration movements of the entire project's worth of mallards. So you can see that across the Atlantic Flyway there are hundreds of transmitters out actively going places, and that's their springward migration. It's a bit hard to see, but that's our mallard locations where they chose to breed. We had one bird from Cape Breton go up just north of Goose Bay in Labrador, but all the other birds bred locally, kind of in areas relatively close to where we marked them. And then our black ducks made a much larger jump. So some of our Glace Bay and Cape Breton birds went on to Newfoundland and the Labrador, and then birds that we marked in Prince Edward Island did quite an extensive migration up in the Labrador. This is Churchill Falls is right here, just as a reference point. So some really cool migrations coming from these birds. Because the information we get allows to understand nesting propensity, so the number of them that choose to breed, of those how many laid a full clutch, of those what we infer are successful. We get some very simple reproductive rates that we couldn't get otherwise from these species. Black ducks nest roughly 1 pair per square kilometer in the boreal forest, and you can imagine in that remote landscape it's hard for us to be out there finding that one nest in that square kilometer and then following that duck through to completion of brood rearing. So this technology is super helpful in helping us to better understand some of that information about these species. And then I mentioned helping with some American woodcock work. There's a large Eastern North America woodcock project being led through the University of Maine. They had a PhD student that was interested in looking at overwater migration of woodcock coming out of Nova Scotia. And kind of all the streaks here are the culmination of all the transmitters we deployed over 3 years, and those migrations over the Gulf of Maine, and also some other strategies of just crossing the Bay of Fundy and then taking a more terrestrial route down to their wintering grounds. The kind of conservation impact that comes from this is that these transmitters give us an altitude estimate. On average, woodcock are doing these water migrations in the fall at an average height of 110 meters and ranging between 10 meters and 294 meters. You can imagine woodcock, which is kind of like the bumblebee idea where they probably really shouldn't be able to fly, but they can, flying 10 meters over the Gulf of Maine. It just kind of is an interesting thought. But anyways, where we currently are in offshore wind industry, the motor sweep area ranges between 20 and 206 meters, so that's not a great height for woodcock. Not trying to infer that there's going to be impacts on woodcock, but it doesn't look really great for these woodcock right here. So there may be some adaptive strategies that woodcock will take over time, but there is quite a bit of offshore wind proposed. That's the U.S. This is the Canadian side. We do have woodcock migrating to Newfoundland and Labrador, so there are potential impacts for that species with offshore wind, just based on the height that they're on average migrating. So something that's coming from this project, a whole bunch of neat movement data and a bunch of cool things, but that's what I've been working on. And with that, that's probably awfully long-winded, but thank you for your patience. There's a lot going on, and we do a bunch of really cool stuff. Any follow-up, there's my email, there's my cell phone. All of you can probably find me far faster through Liza or Jeff and get to me that way, but happy to answer questions or follow-ups through email or cell too. Thank you very much.
Any pressing questions?
What about raptors?
And avian influenza? Yeah, so raptors are ones that through scavenging typically are picking up virus. So where there are die-offs of things like Canada geese and eagles are scavenging on those carcasses, that's something where eagles are picking up avian influenza. Some of the mammal species that I listed, like red foxes and skunks, it's a scavenging avenue for where they're picking that virus up. So they're on the landscape obviously to be cleaning up those carcasses, but that does make them susceptible to the virus. Crows are another example of that, where they're picking the virus up through scavenging and consuming those, but not really spreading them. You imagine the big concentrations of crows, especially this time of year, the large flocks that are overwintering in places, it's not spreading through that flock. We're not seeing large die-offs of crows. It's an endpoint species that they can get that virus from consuming dead infected individuals, but it doesn't go beyond that. Yeah.
Ted, are there other hot spots of avian flu influenza?
Yeah, so across the four flyways, this time of year, people are seeing increased occurrences. I don't want to call them outbreaks, but there's increased occurrences. So we've had some geese that have died in Prince Edward Island. We've had some geese that have died in southwest Nova Scotia as well as in the HRM. And then along the Atlantic Flyway, I just got back from the Atlantic Flyway Council meetings, and state partners in the US have talked about increased occurrences that have been happening more recently, just with kind of the pressures of winter and concentrations of birds, and again, not an unexpected amount. That way, we see these increases in occurrences happening this time of year, and then as the birds spread out to their reproductive areas at the end of migration, it really drops off, and we don't see a lot of occurrences through late spring, summer, and not until birds are coming together again in the fall that we really get an uptick again in occurrence.
Is there any citizen science on this? Like, can an individual take an oral or fecal swab with a Q-tip?
So not directly, no. And I mean, that's just for, keeping those folks and the virus separate. people that are instructed through, you know, provincial avenues or through the Canadian Wildlife Health Cooperative are often asked to, you know, have gloves and a mask, and if not, be using plastic bags to pick up the carcass and then bag again and again, and then that's taken to the facility in PI in our area, where the people in the lab then do that for the monitoring. And it just helps keep people safe, make sure there's no contamination with the sample and that type of thing. So there's not really, other than the reporting side, there's not an opportunity to have citizens out there sampling sick or dying birds. And from the hunter-harvest side, you know, hunters are often not harvesting sickly looking birds because they're harvesting them to want to consume. So we have a normal wild looking, wild healthy acting bird that those people are sampling. Yeah.
So what you're saying is currently, avian flu really isn't a big concern, like everything's on the normal arc?
Yeah, so I don't want to say that it's not a concern. I mean, it obviously is a concern. It's definitely a concern for, you know, commercial poultry operations or people that have, you know, poultry domestics in their yard and those types of things. So there's a need for biosecurity awareness and understand that the virus is on the landscape. But in terms of wild birds and their populations, it's not currently impacting them in a way where we're concerned at the moment for that. And within waterfowl, we know that waterfowl carry these types of viruses. They're maintaining that virus in their populations, but they're able to do so in a way and continue on. So it you know, the level of concern is a little bit relative, but I mean, there is concern out there. You know, commercial production of poultry, whether it's, you know, for meat or eggs, has been significantly impacted in North America and around the world, and that's something I would say is concerning. But for the wild birds that I work with, it's not a concern at this point. Yeah.
If one has a few chickens in the backyard, but you're not close to any water sources and you don't get waterfowl, but you have feeders out, I understand that the feeder birds are not a high risk.
Not to our current knowledge. I mean, for, you know, kind of the backyard poultry folks, you know, husbandry actions and keeping biosecurity is something that's on each individual to do, and the greater and better that you do that, the healthier that your flock's going to remain. You know, there's other things that wild birds carry that isn't just avian influenza viruses that can be transmitted to local backyard flocks, and so keeping wild birds out of feeders and waterers is important. And if they're outside free ranging, you know, being aware of what's in the landscape, and, you know, if you see birds that are wild going into your feeders, I would clean those, and being precaution, you know, approach is really important. And I think if there's more specifics, probably asking a veterinarian is a bit more you know, pertinent than me. But at this time, for, you know, what we're really seeing as kind of source populations and ones that are vectors for the virus, it's typically waterfowl in sort of an urban environment, but gulls can carry that, and gulls are relatively far-reaching, especially in an agricultural or backyard agricultural situation, right? And gulls are going to be maybe more on the landscape in the coming months than they have been recently. So due diligence is important. Yeah.
Well, Ted,
Okay.
thank
Wonderful.
you
Thank
very
you.
much.
Yeah, thank you all.
That was excellent. You guys are certainly keeping busy.
Thank you.
Good job.
Yes. Oh, yes. No. And I mean, it's great. It's nice to be on the landscape and contributing to something that is impactful at a national and international level. And to think that we can be monitoring to that prevalence rate of 1%, I really think is kind of empowering that we're out there on the level that we've got a pretty good understanding of where the virus is at, and you know, being aware that it's there, it hasn't gone away, and we have, you know, lower prevalence than we have in the past, but it isn't gone. So I think that's a good awareness for everybody.
And I'm pretty impressed with some of the technology there. Some of those mapping things, it must
Yes.
be really fun to.
It is,
to
it
do.
is.
Yeah.
They become almost kind of like children you need to watch.
Yeah.
while
Look
they're
at this,
making-
what this guy doing?
Absolutely, and I mean, some of these birds, I mentioned that bird that went to Greenland
Yeah.
and was in urban, Philly, like, it just is sort of interesting how they can see such a change in a short time period, but become accustomed to them
Yeah.
so easily. It's
Pretty
quite impressive.
adaptive.
Absolutely, absolutely. So, yeah,
Good.
really, really neat birds and
Good.
neat to be part of those projects.
Great, well, stick around and contribute, hi Jaden, and contribute to our sightings if you have time or
For sure,
interest.
yeah, no, we're here till the end
Because
of the
I'm
meeting, for
sure Jaden probably saw something interesting today.
Yeah.
Wonderful.
Great, thanks a lot.
Yeah, no, my pleasure. Thank you all.